as I understood, the topic of the day is, this Darwin Day is domestication. So I want to look at domestication from the point of view that Marion Lem and myself have developed in the book. I would just start with saying thank you very much to the people that are responsible for the ideas that I'm presenting. It is only part of the people, but I thank Marion Lem for everything, Eitan Avital for the work we did together on animal traditions, Daniel Dorr for the work we did on the evolution of the linguistic capacity, and Anna, who's sitting here, for her beautiful and original pictures, which make the book so much better and all my presentations so much more beautiful. And I always thank my students for always they challenge me, and they are always curious and always inspiring. So we're going to talk about domestication. And before I start presenting the point of view through which I want to look at domestication, I want to say, what are we talking about? What is domestication? And here we have a kind of introduction, which is fine, and that is the taming of wild animals and plants by humans. Animals are often herded, and their care and feeding becomes dependent upon humans, while plants are farmed and often become dependent upon humans for propagation. Domestication changes the physical characteristics of the plants and animals involved, and it altered the evolution of humans. So we're talking about a process where human beings change the biological world in which they live and make it more useful for themselves. And they change very fundamental characteristics of the animals and plants that they use, and also of themselves as they domesticate them. There are other aspects that I will touch about humans in this respect. So this is an example of domestication that Darwin used, and this is the domestication of the pigeon. And here we have the rock dove, the original, the ancestor of all the many, many strains of pigeons that men have fun in raising and in breeding. You see here, oh, where is it? Oh, never mind. Here it is. Here, the domesticated pigeons, and this is the rock pigeon. Now, Darwin used domestication in the first chapter of The Origin of Species. Also, very interestingly, when he wrote in 1844 the essay, the big essay that was the basis of The Origin of Species, he also, the first chapter was about domestication. So the basic structure, he had it for quite a long time. And what he was trying to do was to take domestication and say, look, here we are, and we're changing animals and plants through all kinds of means, and the change is very substantial. It's a change in the biological properties of these animals and plants, and he focused on animals, he focused on the pigeon in his in this chapter, but not only on the pigeon. And he said, well, we can learn from this artificial selection and from the methods that humans are using about what is going on in nature without the intervention of humans. And he used this model in two ways. The first, he used it as a model for natural selection, because he said, here we have these pigeons, and some of the pigeons are a little bit different from others, and we pick up those that we fancy, those that we like, and we begin to breed them. And then again, we pick up from those that are according to our liking, and so on. And this is selection by men, and there is an equivalent in nature. And we all know this, because this is usually how this is, when we read the first chapter and when we read about the first chapter of The Origin, this is how this is presented to us. But very interestingly, Darwin also used domestication as a model for the generation of heritable variation under extreme conditions. Because Darwin said that putting animals and plants under domestication is putting them under very, very unnatural conditions, very strange conditions, very extreme conditions, socially different from the ones to which they are used. We are giving them plenty of food, which they don't have under natural conditions. We are putting them sometimes in very different environments. This situation of domestication is therefore a very extreme situation, environmental situation for the animals and plants. And this stress, these extreme conditions are causing a generation of variation, of new variation. So domestication is giving us a model also for what happens to animals and plants under extreme conditions in terms of generation of variation. And what Darwin was asking, and what we should ask, I think, with him, is what varies exactly? How is variation generated? Heritable variation generated. What exactly are we selecting, always? And how does selection proceed? At what level are we selecting? What is going on? Are we selecting genes? Genes are selected, but are we selecting them, or is it just a byproduct of our selection, and so on? Of course, he didn't know about genes. So I'm quoting here from the essay of 1844, just to show that what I'm saying is not a figment of my imagination, but this is actually what Darwin said. And Darwin said this: races are made under domestication, first, by the direct effects of external conditions to which the species is exposed, two, by the indirect effects of the exposure to new condition, often aided by excess of food, rendering the organization plastic, and by men selecting and separately breeding certain individuals, or introducing his stock, selected males, or often preserving with care the life of individuals best adapted to his purposes. And three, by crossing and recrossing races already made and selecting their offspring. This is what Darwin said. So here it's very, very clear. It is a model for the generation on variation, and then for the shaping of this variation through selection and through selective crossing. And this is also what he says in The Origin, exactly the same things, in fact, in a less condensed way. I have the quotation here. I will not read it all to you because it's not necessary, but the basics are there. Again, conditions of life have an effect on the biological organization. I don't know if you know, but Darwin was, unfortunately for some, a Lamarckian. He believed that the conditions of life are changing. It can change what he called the change the biological organization and change the heredity directly. And he believed also that use and disuse is important in evolution. Never mind about that, but this is what he believed. And he believed that under extreme conditions especially, these things are very evident. And then also you're selecting, doing the selecting bit, and then you're also doing crossing and recrossing, and all these things together end up in human beings being able to shape the animals and plants around them in a way which suits their interests and their fancy. But he believed, and he said this, I am convinced that the cumulative action of selection, whether applied methodically and more quickly or unconsciously and more slowly but more efficiently, is by far the predominant power, especially for animals. So, although all the other factors are very important, selection is particularly important. And he stressed this very much in The Origin. Okay, now we know more or less what domestication is, and we know what Darwin thought about it. So I want to take now a step back from this and to try and look at this process of domestication through the particular framework that Marian Lamb and I have developed in Evolution in Four Dimensions. And in Evolution in Four Dimensions, what we're trying to say is the following. We're trying to say, look, if you're thinking about the way that information is transmitted from one generation to the next, and how variations in information are transmitted from one generation to the next, we have to consider all the routes of transmission and all the ways in which this information can be transmitted. And DNA is one route, of very important route of transmission of information from one generation to the next, but it is not the only one. And it is a mistake to focus only on this. It is not the bottom line, and we can forget everything else. We cannot forget everything else, because we will not understand also what is happening at the DNA level if we don't understand the other things. So one of the ways of thinking about it is saying, okay, let's think about something that will not be very controversial, about development, about how an organism develops. From, let's say, a fertilized egg, let's think about a human being, from a fertilized egg to an adult. And when we think about a human being that develops in this kind of way, we are thinking, we can ask ourselves, what are the inputs into development? What do we put in? And then we get the adult phenotype. And of course, we put the genetic resources. So let's think in terms of resources into development. What are the resources? So we have the genetic resources. A difference in DNA makes a difference in development. We accept that, we know quite a lot about it. Then we also have other resources into development. We have the non-DNA part of the egg, which is both the chromatin of the non-DNA parts of the chromosomes, and also the cytoplasm. And this also we know that if we change these things, the chromatin, we change the cytoplasm, we can change the phenotype. Then, we're thinking about a human being developing, we're mammals, we develop in a womb, and we know that the environment of the womb is very important for development. If we change the environment of the womb, and I will give you examples afterward, we can change the development of the organism. And also we know that what we're giving very early on, the milk the mother gives, if we're in mammals, is important input into development too. It can make a difference to some aspects of the development of the fetus, of the child. And then we can think about the early environment of the developing human, about home and care, and we know that this has a very great impact on development. And in the end, we can think about the social and cultural world in which we live. And one of the ways, and these are all, we're talking about it as inputs into development, but I want to argue that all these inputs into development are also inputs into heredity. They affect not only what happens now in this generation, in the development of this individual, but can have transgenerational effects. Effects on grandchildren, great-grandchildren, sometimes for many generations. And I want to describe this in terms of a beautiful picture that Anata drew, which is called the 5 mothers. And the 5 mothers that we have here we have the mother that gives the DNA. This is this mother. You see the DNA here? So she gives the genetic resources. The other mother gives the non-DNA parts of the egg. This is the egg. And she gives the chromatin, and she gives the cytoplasm. And then we have the womb mother, and the early postnatal mother, the one that gives the milk. Here we see the antibodies in the milk going to the child. And this affects development. And then we have the great mother, all the other things around us, the great cultural mother, mother culture, that we all are part of. And in terms of these 5 mothers, these 5 types of inputs into development, we can think about what makes a difference in the development of, in this case, a human being. And we can, but I'm saying that these things are also inputs into heredity, because changes in DNA, of course, can lead to changes through the generations, but also epigenetic changes, changes in chromatin, for example, can have long-term transgenerational effects. Changes in certain habits can have transgenerational effects, and of course we have cultural evolution, symbolic evolution, which is part of all of our lives, that of course makes a very big difference to us in many ways. So we are thinking in terms of these inputs. And I will say a little bit about each of them, very little. And then I will go back to domestication to see what role these types of inputs play in the process of domestication. So here we have the DNA. I'm not going to talk much about it. We all know it. It's one of the icons and symbols of the 20th century. And it's very important. And we know that variation in DNA leads to hereditary variations, which are to phenotypic variations sometimes, and this can be inherited from one generation to the next. But we also have something that is called epigenetic inheritance, which maybe you are less familiar with because it's not appearing in all the textbooks. And epigenetic inheritance is defined here as the inheritance of phenotypic variations that do not depend on corresponding variations in DNA sequence and on the persistent presence of inducing signals in the present environment. And it may sound to you as if I'm saying something against the importance of DNA, but I want you to note that what is said here is not that we can have variations that are independent of DNA. We can have variations that are independent of variations in DNA. We can have phenotypic variations that are independent of variations in DNA. This means that we can have, for example, two individuals that are genetically the same, that live in exactly the same environment, and nevertheless have different characteristics, and these characteristics are transmitted to their descendants. And the reason that they are different is because sometime in the past, something happened to one of their ancestors, and this something has changed for example, patterns of gene expression, and this have become inherited from one generation to the next. And what has happened during the last 20, 25 years is that we understand the mechanisms, the cellular mechanisms that underlie this type of systems that allow this kind of thing to happen. I will not go into details here about these biochemical mechanisms. They are described at length in this book, but it's a little bit technical sometimes. But I want just to say that unlike the case in DNA, when we have DNA replication is a basic mechanism, one basic mechanism that, although very complex, that we have to think about, when we're thinking about epigenetic inheritance, we're thinking about several mechanisms that are quite different, and we have to always ask ourselves what is going on, which mechanism is working, how is it working exactly, what is the nature of it. It's not given. We have to ask ourselves this question and find out. If we have an epigenetic phenomenon, we don't immediately know how it is transmitted from one generation to the next. We have to do quite a lot of work in order to find out. Sometimes it's easy, sometimes it isn't. And I will not go into all the mechanisms, but I want to just to point out that there are self-sustaining loops, metabolic loops. There is structural inheritance, three-dimensional kind of templating. Those of you who know about prions, this is how prions are propagated from one generation to the next. There are things that happen at the level of chromatin on the chromosomes, and there is a Rna Mediated Inheritance, where RNA is very, very important as a source of both regulatory changes, which are then inherited, can be inherited from one generation to the next. The other thing about these systems, which is different from DNA inheritance, is that with DNA, once you make a change, and this change stays, it's not repaired, for example, but it stays, that's it. It goes on. And it doesn't very much matter what exactly the conditions of life are. With epigenetic inheritance, the very fact of inheritance is it is induced under some conditions, and what conditions it is an epigenetic difference is induced in, for how long, for how stable it is, are not given. Again, this is something we have to study and find out. So it's quite a complex system. And it's a complex system because the epigenetic changes are both regulatory developmental changes and hereditary changes. It's like playing a double game. It's always very complicated. So I'm not going to go into this, but this is one chromatin marking system that we understand in some molecular detail. This is the system of DNA methylation. And DNA methylation is a modification of DNA where a methyl group, the little creature up there, this and this, is tied up with a cytosine that is near a guanosine, CG, and the patterns of methylation, and whether or not a gene is methylated in the CG sites makes a difference to its potential of being transcribed. And the point is that patterns of methylation, different patterns of methylation can be imposed on the same DNA sequence, and once a pattern is imposed on DNA sequence, it can be inherited from one generation to the next. And this is a very, very important part of chromatin marking epigenetic inheritance, although it is not the only type of chromatin marking epigenetic inheritance. As I said, I don't want to go into details about the more technical parts of biochemistry, but the systems are now understood in some detail, and they are not anymore something, very baffling for us. I mean, they're just biochemical mechanisms, important biochemical mechanisms that we understand, and we understand them as memory systems, kind of cellular memory systems. Usually we think about them in terms of cell memory in development. For example, different cells in our body which have the same DNA have different phenotypes. They also have different methylation patterns, for example, and so on. So we understand them, but it turns out that these patterns of methylation are not only making cells different from each other, they can also, differences in methylation can also sometimes be transmitted from generation to generation and can lead to changes in the phenotypes of descendants. And here we have an example of an epigenetic difference between two animals. The big, huge, fat mouse and the little brown mouse have exactly the same DNA. And they are living now in the same environment. They are eating the same food. They are living in the same cage and so on. The differences between them, they are genetically the same, yes? It's a pure line. The difference between them is the environment they experience in the womb, what happened to their mothers. The mother of the little brown thing was eating a diet full of folate and methyls, and the other mothers didn't. And the mother that had a lot of folate produced a normal brown mouse, and the mother that didn't have folate in the diet, in this particular line, produced the huge, monstrous yellow mouse, which is also rather sick. It has diabetes, tendency to cancers, all kinds of nasty things. And what is very interesting about this is that a brown mother, her offspring will tend to be more brown. The offspring of the yellow mother will tend to be more yellow. So it's not only that this is their phenotype, which is different because of the environment in which they developed, this have effects on the next generation as well. And here is another example. Again, these two plants are genetically exactly the same. They are living in exactly the same environment, but they're different. The morphology of the flower is very, very different. And it is different not because of a change in DNA, but because of change in DNA methylation, an epigenetic change. And this is transmitted. So the normal flowers here, these are the normal flowers. A plant like that will produce offspring which will also be normal, and this flower will tend to produce offspring which are abnormal, like itself. And sometimes we find in plants some flowers which are intermediate between these two forms, and when we look at the methylation in one of the major genes that are controlling flower morphology, we see that the level of methylation is also intermediate. This is a very interesting flower. This is Linaria Vulgaris, and it was a plant that was studied by Linnaeus. and made Linnaeus very worried about the origin of species. And that's why people have looked at it for historical reasons, because, you know, it was some years ago, it was 250 years to Linnaeus, and Linnaeus' day was coming, so people were looking at Linaria, and they thought, well, we have the gene for flower morphology, let's have a look at the classical flower, at the classical species that Linnaeus was looking at, and they were expecting to find a mutation. There was no mutation. The DNA was the same sequence, no difference. So they didn't, but then they looked at the methylation pattern and they saw that this is what is different. This is what makes this flower different, and it is making them not just different, it's making them heritably different. It's a very, very, it's a very interesting, somewhat ironic story. And here we have another example, which is, as I always, since it has appeared in the literature, I talk about it because I think it's very, very important. This is the story of the rats that were giving vinclozolin. Vinclozolin is a fungicide, and this fungicide is when an injection is given here to the mother, and this injection is leading, here we see the mother, and she has an embryo, a male, who is developing inside her, and she's getting an injections in days 8 to 15 of gestation. It's very important because this is the time where the germline of the offspring is developing. And what happens as a result of this injection is that the little male that is born to her is rather sick. It has a testis disease. Its offspring also have this testis disease, and the great-grand offspring also have the testis disease, and so it goes for 4 generations. One injection to the mother, one. And it goes for 4 generations. And what we know about this system is that they isolated about 15 different DNA sequences that have altered methylation patterns. And these methylation patterns are reappearing in every generation of sick rats. Now this is important because vinclozolin is a fungicide that we're all using on vines. And although the concentration that was used on this female is very high, we don't know exactly, I'm not sure it's very good for the human beings in small concentrations over a very long time. So it's something that we should really look very carefully at. In general, we have to think about all kinds of chemical pollutants, not only in terms of what they're doing now, but also in terms of their possible transgenerational effects, which can be very serious, as we see from this experimental example. Now here, as I said, I was talking before about changes that happen in animals as a result of epigenetic changes in DNA methylation I was talking about, and these changes go through the germline, through the egg and through the sperm. But we can also change the characteristics of animals by changing in a systematic way their development, and this can be transmitted from one generation to the next, bypassing the germline, not through the germline. Nevertheless, it is important. It is a transmission of information. And I want to give you, again, an example that was studied by Mini and his colleagues in Canada. And here we have a rat. And this rat is licking its offspring, and the offspring usually a rat licks its offspring a lot, and well-licked offspring are brave little rats. They go, they explore the world, they are quite healthy, they are not stressed very easily, and so on. Sometimes we have a mother that is not licking her offspring a lot. She is licking them, they are clean, but not enough. But they're not licked enough. Now we know that licking is very important in the maturation of the nervous system, for the normal maturation of the nervous system. And what is interesting is that the offspring of a female that are not licked a lot, that are poorly licked, develop into cowardly little rats. They are afraid of everything. They don't explore their environment much, and they're stressed easily. When you stress them, the corticosteroids jump a lot more than the other rats. And what is very interesting from our point of view, from my point of view, is that the female rats of this mother, the little daughters, when they become mothers, they also don't lick that much their offspring. Part of the pathology is that they also are not very good mothers. So this is perpetuated in the line. So again, we can have two rat lineages, which are the same genetically, living in the same environment now. One is stressful, cowardly rats, while the other one is little brave rats. And they have the same DNA, they are in the same environment, but something happened in the history of the mother which is getting perpetuated from one generation to the next. And I must say that it looks a little bit like abuse that runs in families. And in fact, there are monkey models for this kind of things too. Not quite similar. So here we are. And this is a picture that is showing us another type of transmission of a tradition in chimpanzees. And we have two types of, two chimpanzees here. One is opening nuts. The other one has a way of fishing termites. And they are transmitted by social learning from one generation to the next. And we know that it is social learning. It's not that one has genes like this and the other one has genes like that. The genes are the same. And we know that because we can do cross-fostering experiments, and also because we know that there are environments where there are nuts, but the chimpanzees don't have a clue how to open them, and they don't even try because they don't know that they can be opened. So it is something that somebody learned at some point, and we know, by the way, or we suspect, that opening nuts was learned at least 400 years ago from humans, because there are some reports by missionaries about chimpanzees opening nuts 400 years ago already. And one of the suspicions is that it happened in chimpanzees that were close to human habitations. They learned it from humans, and then they transmitted, and since then, for at least 400 years, they're transmitting this. And we know about this kind of thing quite a lot. And now we're coming to our culture. It's the symbolic culture. We live in a very, very special world, which is rather different from the world of other animals, although we share with other animals all the other dimensions of heredity. We live in a symbolic world, in a symbolic landscape, which we see here. And this lecture is part of this, and this museum is part of the symbolic landscape. Now we go back to domestication. And here we are, and we see these dogs that have all, as far as we know, evolved from the wolf, or some wolf-like animal. And look how different they are. And this caught the attention of the first of great evolutionary biologists. Lamarck noted it and wrote about it in the Philosophie zoologique, and so did Darwin. Here we have, again, dogs in our lives. Yes, you can see the relationship between this boy and the dog. Very strong relationship, obviously. And you see another relationship between working dogs and men, or woman, I'm not sure. And what we want to ask is, I'm just going to Lamarck just to give you an indication that here is a quotation. Lamarck said, "Where do we find those bulldogs, greyhounds, water spaniels, leopards, etc., races which show wider differences than those which we call specific when they occur among animals of one genus living in natural freedom?" He said, the variation, the morphological variation is enormous. It's more than between normal species. Where does it come from? No doubt, he says, a single original race closely resembling the wolf, if indeed it was not actually the wolf, was at some period reduced by men to domestication. And then he suggests what happened. He thinks that it is an example of the inheritance of acquired characters. For Lamarck, this was his way of thinking about this. So he said that race, of which all individuals were alike, was gradually scattered with men into different countries and climate, and after they had been subjected for some time to the influence of their environment, of the various habits which had been forced upon them in each country, they underwent remarkable alterations and formed various special races. So for Lamarck, it is the conditions that molded, that changed the animals. Because they were in different conditions, they have changed. And Darwin writes, here we have from the essay, the 1844 essay, which I already read to you, but if you read this bit here, he said, looking to the domestic dog of the whole world, I have, after laborious collection of all known facts, come to the conclusion that several wild species of Canida have been tamed and that their blood, in some cases mingled together, flows in the veins of our domestic breeds. This was one of the things he said. He thought that it is not one origin, it's not the wolf. He thought also that the jackal probably also was an input into the modern dog. And then he says, again, he's talking about the dogs, and he thinks that for who will believe that animals closely resembling the Italian greyhound, the bloodhound, the bulldog, pugdog, etc., so unlike all wild Canida, very similar to Lamarck, ever existed in the state of nature. It has often been loosely said that all our races of dog have been produced by the crossing of a few aboriginal species, but by crossing we can only get forms in some degree intermediate between their parents. And if we account for our several domestic races by this process, we must admit that former existence of the most extreme forms as the Italian greyhound, bloodhound, bulldog, etc. in the wild state. Moreover, the possibility of making distinct races by crossing has been greatly exaggerated. And he concludes by saying it is natural selection mostly. Yes, crossing adds to the formation of new breeds, but it is mostly natural selection of those variants that man has decided to choose, and the systematic choosing of this every generation of the variants that one is interested in, that is leading to the very many races of dogs. Now, what do we know? Dogs are studied quite a lot, and the evolution of the dog from the wolf is a topic of quite a lot of research, mainly at the DNA level. But I want to look at this animal here that you will see. This is a fox, a silver fox, a domesticated silver fox, and I want to tell you about a very interesting experiment that has been done. But before I describe this experiment, I want to say one thing. Obviously, when we're selecting and crossing, we're selecting variations, phenotypic variations, and obviously some of the variation is genetic. There is no doubt about it. So we're selecting for DNA, but are we selecting only for DNA? When we're selecting a phenotype, what exactly are we doing? And the work, and how are we selecting? I will come to that soon. How exactly are we selecting? It is very important to understand this in order to understand the process whereby we end up with a particular breed which is genetically stable. And that's why the greatest experiment on domestication in animals has been done on the silver fox. It has been done in Russia, in Novosibirsk. And it is now 46, and it started in the end of the 50s, in the end of the 1950s, by Belyaev and his group in Novosibirsk. And what they did was they took the silver foxes, which were bred in farms in Russia for about 100 years, and they were bred for their fur. So you breed the foxes, and then you kill the poor things, and you take the fur. And Belyaev thought, when he was in Russia, before he went to Siberia, when he was looking at this fox farms, and he thought that trying to domesticate these foxes could be a very interesting experiment. An experiment that can tell him a lot about evolution, because he was very deeply interested in evolution, and also an experiment through which he can convince the authorities to give him a little bit of money and freedom, because of course if you can domesticate foxes and make them more tame, it's good for the business. So he started the institution of he was heading the he has become a head of the Institution of Genetics in Novosibirsk, and he started his experiment in the end of the 1950s. And the experiment has been going ever since, and what he started doing was he started to selecting for tameness, for foxes that were not very aggressive, they wanted human, that at first were not very aggressive, and then those that were actually keen for human touch, that wanted to, like dogs, that wanted to be with humans, that not only did not bite them, but wanted their company. Now, he did this in a way that was very, very sharp, in a sense brutal. He just put a handed glove in, and he saw the reaction of the foxes at particular times to see whether they withdrew with fear and snarled at the hand, or whether they approached it and were curious about it and sniffed it and so on. And this was the process of selection. These are the baby foxes. And things started happening very quickly. He was very successful in this experiment. He started, by the way, an opposite experiment 10 years later, selecting for aggressiveness. So now there are 46 generations of foxes that are selected for tameness, and there are 36 generations of foxes that are selected for aggression, and in both cases, very, very successful. But I'm going to focus on those that are selected for tameness because we're talking about domestication. Things started happening to these foxes which were not anticipated. When he was selecting the foxes for tameness, suddenly a lot of foxes with these markings appeared. These white markings that you see here are not normal in wild foxes. They don't have them. It's called white spotting, and I will talk a little bit about this kind of thing because this is behaving like a dominant or semi-dominant character. It's appearing at high rates. It is reversible. It doesn't behave like a genetic mutation. It behaves beautifully like an epigenetic variation, although we don't know exactly, we don't have the data at the molecular level yet. I hope that we shall have soon. Other things happen to the foxes. Here they are. You see the tail here? This is not a normal fox tail. This is a dog tail going up curly. This is not, there were changes in, you see here the spots, but you see also, this is a normal skull. This is the skull of a domesticated, of a domesticated fox. There are morphological differences in the skull. There are differences in vocalization. Some of these foxes even started to bark. Not many, but there are very important and very obvious differences in vocalization. A whole host of characters in a coordinated manner has changed. Not one, not two, many, and quickly, very, very rapidly. What is going on in this kind of thing? Now, the other interesting thing is, if you are looking at all the domesticated mammals, the cows, the horses, sheep, you name it, dogs, cats, you see in many cases very similar phenomena. The other thing that happened to them is their ears drooped. Many of them suddenly their ears dropped, like in dogs. Now all these things are happening in all the domesticated mammals. There is a whole series, a whole set of characters that when you are selecting apparently for tame behavior, and this is what humans have been doing with their dogs, with their cows, and so on, they're not selecting like Belyaev was selecting. They were selecting in other ways, more complicated ways. But they were selecting for this. They changed something that led to a whole set of characters changing in a coordinated way, and the same type of characters, which is a very, very interesting phenomenon, and Belyaev had an explanation for that. Now, here is a whole set, a summary of all the set of characters that have changed as a result of domestication of the fox. And also the other thing that is very important that I didn't talk about was that the loss of the seasonal rhythm of reproduction. They have already females coming into heat twice a year rather than once a year. And this is also something that has happened, of course, with the dog that they're not breeding like wolves just once a year, but they're coming into heat a lot more often. Okay, now what is happening? So what Belyaev said was this. Obviously, we're targeting development. And we're targeting, I will talk about this picture very soon. But what we're doing here is we're targeting the neurohormonal system. The nervous system and the hormonal system and the relationship between them. And we're targeting it during early development. And that is why it has many, many effects on the phenotype. Now, what he thought was happening, and from this point too, he was a pioneer, he thought that what we were doing, we were not only selecting for particular types of genes, obviously we're doing that, but we're also selecting for those genes that can have epigenetic transgenerational effects, and we're selecting for, very often we're selecting for epimutations in these genes, not genetic variations. For example, in the case of the white spotting, it seems like this is an epigenetic variation rather than genetic variation. Now, we don't know a lot about it, that's why I'm very hesitant about these things, but it seems to me that when we're selecting, for example, for tape behavior in animals, we're selecting all the heritable variation that are giving us the result that we want. And these are genetic variations and epigenetic variation, and the indication that they are some of them are epigenetic is given to us by this white spotting, the behavior of this white spotting character, which is really not complying to any classical Mendelian character, and we cannot give the usual genetic explanation of inbreeding because there was no inbreeding there. They made sure there was no inbreeding, and no mutation pressure at this level is impossible. It's too common, and so on. So it's very interesting. Now, what we see here in this picture, we see the wolf and the domestic dog, the wild silver fox, and the domesticated one. And I will come to the other ones too. And we are obviously selecting for tameness here. We're selecting for changes in temperament, for animals which are less fearful, for animals which are less aggressive than the normal wild animal. And what is very interesting is that the change in aggression and the change in fearfulness, when you are selecting for a temperament which is more docile, you're also selecting for attention, social attention, because an animal that is in a social contact with another animal, it's very, very nervous, and it's very fearful and very aggressive. I mean, it will not try to cooperate and communicate very much in a kind of friendly way with the other animal. All it's worried about itself, it's worried about a possible attack, and so on. So it is quite possible that by selecting for a docile temperament, we're also selecting for the ability to learn and to cooperate with other individuals. And this possibility was checked in dogs. Now, one of the things when you're looking at the dog and the wolf, you see that the dog has lost quite a lot of its brain during domestication. Dogs have about 30% of the size of the brain of a wolf. And wolves are very, very clever animals. But they're not very good, for example, in some things. They're good in a lot of solving a lot of problems, non-social cooperative problems. But they're not very good at, for example, understanding that when a human being gazes at some point, looks at something, it means that this could have some meaning, and they can follow this gaze. And on the other hand, dogs are extremely good at these things. Dogs learn this very, very quickly. Dogs are very good at solving social problems which need attention and cooperation and sharing of information. Wolves are not good at this. They are good at other problems, ecological problems. But this type of problems they are not good at. It's very, very interesting. Now, the wolves also are good at, for example, the gaze experiment. They also follow the gaze of human beings. They are also good at paying attention and communicating with human beings. So selection for tame behavior led to a difference in temperament, which led to a difference in the focus of learning. And when we're thinking about ourselves, about human beings, we also ask ourselves, well, human beings are extraordinarily sociable animals. I know that we're all rather cynical about ourselves as human beings, and we have good reasons to be cynical, but because we're a rather nasty species in many ways. But on the other hand, we are a very remarkable species. We're extremely social. We're extremely cooperative. And this cooperation is maybe there was a kind of self-domestication in human beings, which also selected for a temperament which is less aggressive and less fearful than in our ancestors, which enabled us to be to communicate with each other in a better way and a more cooperative way. And again, it is argued by by Tomasello's group, for example, in Germany, that this could have that this could be quite an important factor in the evolution of humans, this selection for a more tame, in inverted comma, temperament in human beings. And one of the things that they have been doing, they were comparing the bonobo chimpanzee and the common chimpanzee. And the bonobo is a lot more, they live in bigger groups, they are less aggressive animals than the common chimpanzees. It's not that they are angels, they're not angels, but they are less aggressive than the common chimpanzee and much, much more cooperative and towards each other. And it seems that, and it is argued, that the bonobo can therefore solve and attend to communicative tasks better than the common chimpanzee. There are experiments that are indicating this. They're not doing as well as humans, of course, but still there is a tendency in this way in the bonobo. Now, so what did we do? What are we selecting? We're selecting genes. We're selecting epimutations, probably. But we are doing it through the selection of behaviors, through selection of behavior that has a lot of effects on many aspects of the animal. We are selecting for the behavior that pleases us, and this behavior has many developmental effects. We are selecting for a developmental system which affects behavior and affects many other things at the same time, because the kind of we are always selecting for a complex of traits. We are interested in one trait, but we can never select just for this trait. We take the whole bundle of traits together because we want one thing out of this bundle. But it is given to us as a whole package, as a package deal. And we are selecting, when we're thinking about behavior, when humans select, they select for tame behavior and for all kinds of other traits which are useful for them. And they're selecting for, and they're doing it through selection on the development of the neurohormonal system, because this is the system that controls behavior. And they're selecting for traits that are important for them. They want the animals to breed often. So they're selecting for this trait. They want animals to breed as they like, not as the animals like. They don't want, for example, the dominant structure of wolves. They don't want just one pair to breed. They want all their animals to breed. So they are breaking the hierarchical structures that exist in animal societies. They are changing the social interactions in animal societies. The dog is the only animal, is the only canid that does not have paternal care. All the canids, all the fathers in the canid family look after their young, except the dog. Why? Obviously, because the dog, the male was not an integral part of the, what men did was selecting for bitches that give birth, and it didn't select for dogs that were looking after pups, he was moving them away. So this trait was lost. And maybe it was also a byproduct of the selection for other things, but this was one of the things that happened during the domestication of the dog. So many, many things happened during this process of domestication. And just looking at the genetic level and saying, this gene changed, A, B, C, D, this 15 genes changed, without understanding what was selected, how it was selected, how it came about, we will not understand very much. We need to understand all the aspects that are partners in this big game of selection. The genes, the epimutation, what developmental system exactly was selected, what aspects of behavior were selected and were targeted, and so on. What kind of repercussions did it have on the system as a whole? How did this thing work? And here I'm giving you just a very short example. We have here this is the jungle fowl, and we saw them in the first, the jungle fowl, we saw them in the first picture. And this is the domesticated species, one of the domesticated species of chicken, the white leghorn. And a very interesting experiment was done recently in Sweden about this. What they did was they took the jungle fowl parents and they stressed them. They put them in an environment where the light and the darkness were alternating, and they really don't like this kind of stuff. And when you put them under these conditions for a long time, for some days or weeks, then afterwards you ask them to solve some kind of learning task, they're not very good at it, a spatial learning task. They really got very thoroughly confused by all this stress, and they don't learn very well at all. Okay. And then they looked at the offspring of this, the little chicks. The little chicks were not stressed. And when they looked at the little chicks of the jungle fowl, they were okay. Their parents couldn't learn, but they can. That's fine. They were not stressed. They can learn. They did the same experiment with the white leghorn, and the results there were different. The parents were stressed. They couldn't learn. I mean, they had problems in learning, but their chicks too were also affected. They also could not learn very well. Now, this is not genetic, obviously. They looked at the eggs to see if the amount of hormones in the eggs was changed. They didn't find a change. I'm not sure they can rule it out completely, and nor are they absolutely sure. But they found a change in the expression of 30 different genes in the white leghorn, and the same 30 genes, the expression of the same 30 genes was also changed in the offspring. And they suspect that this is an epigenetic change which was happening. Now, I think that that one of the things we human beings do, and they don't think about it in terms of very consciously, but they want chickens, for example, that will behave like they want, that they will learn what they want, and that what they learn from them, or not learn from them, will stay with them for a long time. So this kind of variation, learning variation, was probably selected in the white leghorn. On the other hand, it could well be that it is a byproduct of selection for other things. We are not sure, I'm not sure about it, but it can work both ways. Or both things can be true. So here we have a little bit of domestication. And I want to say something about domestication of plants. I was talking about animals, but plants are very, very important, and without the domestication of plants, we will not be here today. And we have domesticated plants for food, and for taste, and for beauty, and for fun, for everything. And with plants, we can do things we can't do with animals. We can create new types of plants, and then we can breed from them by cuttings, by asexual propagation. We can't do it with animals very easily. Here we have some of the plants that we have domesticated. Here we are. And when we're thinking about the domestication, one of the very important things, crops that we have domesticated is, of course, wheat, the source of our bread. And this happened when, through the hybridization of certain species of ancestral wheat that have created the domesticated wheat. And what is important here is that, and it is very often that human beings are taking two closely related species, hybridizing them, and then doubling the number of chromosomes. And when they are doing it, it is not just that they are amplifying the amount and variety of DNA in this species, they're also causing a very big revolution in the whole organization of this new genome that they have created. And the revolution that they are creating is both a revolution in terms of what happens to DNA. Some sequences of DNA get lost, some are amplified, some are rearranged. And also in terms of epigenetic, there are very, very massive changes in methylation during these processes of hybridization and amplification of genomes, which we are doing very regularly to our plants when we are domesticating them. We are crossing a lot. And we know that in the and of course we are selecting a lot also. But we know that during the domestication of, let's say, the vine or the olive, plants which for us here in the Middle East are very, very important, we know that both hybridization and selection were very important in the process of domestication, that there was hybridization with local varieties of the wild type vine and the wild type olive, and these processes of hybridization and then selection and hybridization and selection and sexual propagation and then again crossings with local varieties have added a lot, have contributed to the end product that we see. Of course, in this case, this has not been reconstructed to the same extent that it has been reconstructed in the case of wheat. In the case of wheat that I showed you before, that I showed you here, that I'm showing you in this slide here, the process of what we can reconstruct the whole process by doing it in the lab, by doing the crossings, amplifying, doing the crossings, and so on. And we can see in this case what is happening at the genetic and the epigenetic level. And we're beginning to understand these things. But it is more than conceivable that this kind of things have been happening in all cases. So here we are with our domesticated plants. And this is one of the effects of what is happening here, is that as a result, I want to go back to Darwin and to his suggestion that during the process of domestication, this is a situation of stress. It's an unnatural situation for the animal or the plant. And during this, and here I'm looking at plants, and the unnatural situation can be of many types. It can be an ecological situation of stress. It can be a very small population size. This is also a cause of stress, both genetic stress and social stress for animals. It can be a stress that is due to the amplification of the genome through polyploidy, hybridization, and so on. Now, all these things are not only causing new selection pressures, but they're also causing a burst of new variation. And this variation is then selected. So domestication is a model not only for what is happening for selection, it's also a model of variation under stress. And this kind of variation is very important for the important issue of speciation, the origin of species, because a lot of species originate in conditions of stress. Yes. Now, I want just to finish about humans, just to say one sentence about humans. If we're thinking about human beings, we know that domestication has changed us. It changed us in many ways. It changed our genetic our genetic constitution. The domestication of the cow led to the use of milk. The use of milk led to the selection of the lactase gene, of the ability to break up lactose in the adult. The agriculture and the use of starch led to the amplification of amylase 1 gene, salivary amylase 1 gene, which breaks starch. This was an amplification that happened during the last 10,000 years. Domestication, once domestication happened, it affected the history of humans, it affected the spread of humans across the globe. And Jared Diamond argues that this explains the patterns of dominance and domination in human populations. And if we're thinking about what happens during domestication, domestication may a process which is similar to domestication may have happened during our own evolution, changing our temperament, making us more communicative and more cooperative, and leading to the ability to share information. And here we are. And this is our friend Ipra Mistabra, the one who asked the questions, and the opposite conjecture. So thank you very much.